Good afternoon. My name is Melissa Mann, and I am the president of the SSR, the Society for the Study of Reproduction. On behalf of the Society for Reproductive Biology, the Society for Reproduction and Fertility, and SSR, I welcome you to this medal and lectureship in honor of Roger Short. Roger Short was described as a great man of science. He was a truly inspirational reproductive biologist, and his impact is very broad. So instead of studying maybe one or two organisms, a few of the organisms he studied included the badger, the camel, apes, elephant, horses, hyenas, the red deer, sheep, wallaby, and humans. His interests were wide. He was interested in sexual behavior, sexual dimorphism, sexual differentiation, and sexual selection. In addition, he worked on ovulation, hormones, diapause, sperm, as well as he held an interest in contraception, HIV, breastfeeding, and world population. Moreover, he was highly supportive of early career researchers. So this award is a prestigious international award that recognizes early career investigators who possess the same spirit, creativity, and imagination of Roger Short. They have made outstanding discoveries in the field of reproduction. And the 2025 award winner is Dr. David Pepin. He received his PhD from the laboratory of Barbara van der Heijden at the University of Ottawa. He then went on to his postdoctoral studies at MGH, Massachusetts General Hospital, with Patricia Donahoe. He currently is an assistant professor at the Howard Hughes Medical School and associate director of pediatric research laboratories at MGH. He was the 2022 New Investigator award winner for SSR. And in 2023, he gave a lectureship at the ASRM on innovation in medicine. He's been highly prolific with over 60 publications, 3 patent families, as well as training over 55 early investigators. So today, Dr. Pepin is going to receive the medal and award and then deliver his lectureship on MAH and follicles, fertility, and felines. So I welcome Dr. David Pepin.
Thank you.
So I invite Dr. Bruce Murphy to come up to say a few words.
You did a nice biography. You did a nice biography of David. I'll just add a few points. I've known David. Actually, I first met him in Japan when his supervisor, Barbara van der Heijden, had a collaborative project there, and he spent some period of time there. And it turned out it was, from my view, it was almost a career ender for him, because at the conference that we had there in Japan, 2 of us, Dr. Ben Tseng and I, were were videotaped by David dancing on the stage, and that videotape appeared on YouTube. And I thought, well, fortunately, we're not vindictive about it, so we let him continue with his career. But it's been very interesting, and it was not only a pleasure but an honor to be able to nominate him for this award, because I have watched him first as a graduate student, and where he did a spectacular thesis with Barbara van der Heijden. And then, as you will see in the work that's coming up, is his incredible discovery of of how AMH can affect the ovarian populations. So I won't say any more than that, except again, it's an honor and a pleasure, and I'm sure you will enjoy his discourse.
Thank you. Thank you, Melissa, for the kind introduction. And I guess thank you, Bruce, for your fine words. So these are my disclosures. So I'd like to first thank all three societies, the SSR and SRF and SRB, for giving this prize. It's very humbling. And it allowed me to learn a little bit more about Roger Short. So I've never met Roger, but several people contacted me to tell me their stories about Roger. And there's one that I want to relay because I thought it was a funny anecdote. I have a colleague who was a grad student at the same place, in the same department where Roger was the faculty. And he said that Roger would ask always these left field questions because there's such a breadth and depth of knowledge. And that his supervisor would threaten him that if he slacked off, he would put him on this thesis committee. So it just shows you, how much of a man of science Roger was. And it was a pleasure to read on his discoveries. And although I can't compare mine, to his contributions, I did find some affinity with his childhood. Turns out that he was a very precocious naturalist, and he loved going in nature and watching animals and plants, and he's an avid fisherman. And I found I had a very similar childhood, where I grew up next to a national park and spent all my days outside, looking at plants and animals and fungi. And then I want to, of course, thank Bruce for nominating me for this prize. And also the supporting letters from Rich, Joni, and Barb. You can't find a group of individuals quite as nice and kind as those three. And Bruce, I've known, as he's mentioned, for a long time. In fact, he was a co-author on my first paper that was almost exactly 20 years ago. The other thing, people that I want to thank are basically those that made all this possible, without whom I wouldn't have this prize, and are day in, day out in the lab. Basically, are responsible for every discovery I'm going to talk about. Of course, I mean the model organisms. And just like Roger, we're using a number of models, and I want to, I want to highlight the fact that none of the findings that I'm going to talk about today would have been possible without these animal models. And of course, I have to also thank the humans responsible for some of that work, the lab organisms. I have a terrific team that I'm very proud of, that are very collaborative, and it's such a true pleasure to work with these. And then finally, the more domestic organism, and my wife Laura and my two kids, Isaac and Mila. So I always try to see if I can use AI to try to do the slides for me. And so I asked ChatGPT, what do we work on? And apparently we work on AMH. So they got that pretty much right. And who do we work with? And it spit out this thing. So apparently we've collaborated so far with over 450 people, and on a number of different projects. And this is a bit unwieldy. And unfortunately, I don't have time to thank every single person that contributed to our findings. But I do want to put in a few people here that are directly relevant to some of the work that I'll be presenting today, including my former postdoctoral supervisor, Pat Donahoe. So what do we work on? I'd like to put a little bit more subtlety. I don't necessarily think about AMH as being the prime thing that we study, more that we're an ovarian developmental biology lab, and AMH is a tool that I was asked to probe at how the ovary works. And it's a great and powerful tool that allowed us to discover many great things that I want to talk about today. And we're primarily a curiosity-driven lab. We look for interesting questions and we try to answer them. But at the same time, we're funded by the public, and we sit right here in the hospital. And so when we do find discoveries, we really strive to try to use them to do good, to try to develop treatments and find some clinical utility. And so just as much as we're a developmental lab, we're also a drug development lab. And we've worked in developing gene therapies, recombinant proteins, and small molecules. And principally in 5 different areas: contraception, oncofertility, assisted reproduction, ovarian aging, and ovarian cancer. And today what I thought I would do is give you vignettes on all 5 of these areas of projects and papers that I thought were interesting, and to give you a narrative overview of how we came to these conclusions, because we're always used to seeing these polished slides and perfect stories as though we know exactly where we're going and we know exactly what we will find. But that's not really how science works, right? It's finding things that don't make sense and changing directions and hitting walls and thinking and coming back. And so I'll give you more of the true life how we came about these conclusions. So one of my one of my mentors, Bob Weinberg, always says that the most important thing in science is choosing the right questions. And there are 2 principal questions that we've been interested in the ovary. And one is, how is follicle quiescence maintained? And the ovary is really an extreme example of developmental biology. You have to think that the ovary of a girl is full, it already has the entire ovarian reserve, all the eggs it will ever have, while it's still a fetus inside her mother's womb. And that these follicles can stay dormant for up to 50 years, and then one day decide to activate and grow. And they'll grow 10,000-fold, then ovulate and give rise to a totem-potent cell that can give rise to an entire individual. There's nothing as beautiful as that. And so that's such a profound question to be studying. And it's not a single cell too, it's a full follicle. And so to do this, the oocyte has to talk to the granulosa cells, and they have to do this in a coordinated fashion. And how they talk to each other and how they decide to do this, I think, is a fundamentally interesting question. Another question that we're interested in addressing is, how do follicles change over time, and how do they contribute to ovarian aging? What happens during perimenopause and menopause? And menopause is a particularly human concept. It's not replicated in many different animals, and it remains somewhat of a black box. So how do follicle dynamics impact the ovarian reserve? How do they impact the ovary itself, the stroma, and hormonal function? And why is this getting dysregulated? Why do we get an accelerated depletion? Are also interesting questions that are fundamental to the lab. So I'll give a brief history of AMH, so that we know what was done before. And the story starts with Alfred Jost, who was a surgeon in Paris. And he was interested in hormones that control sexual differentiation. And the way he would do this is these very delicate surgeries where he would transpose gonads between male and female fetuses in pregnant rabbits. And so if you put the testis in the place of an ovary in a fetal female rabbit, what ends up happening is you get regression of the Mullerian ducts. The Mullerian ducts are basically what gives rise to the oviduct, the uterus, and the cervix. And so there had to be a hormone produced by the fetal testis that would cause regression of the Mullerian ducts. You next put in a crystal of testosterone and showed that testosterone could not recapitulate that function. So there had to be another hormone, what he called the hormone inhibitrice. And it took until 1986 for this hormone to be cloned and purified by 2 teams across the Atlantic, Nathalie Josso in Paris, which I actually trained under Jost, and Patricia Donahoe here at MGH. And they were able to clone the gene and show that it was actually a protein hormone that was produced and caused regression of the Mullerian duct. And Josso called it MIS, Josso called it AMH, and Pat called it MIS. So just to summarize this, the fetal testis produces AMH, causes regression of the Mullerian duct so that males don't develop a uterus, so that you have proper male sexual differentiation. In the female, in the absence of AMH, the Mullerian structures develop. But it was quickly realized that actually this is also a female hormone, just the developmental timing is different. It is expressed later on by the ovary, and it's expressed particularly by growing follicles, mostly secondary and small antral follicles. And so this was immediately clinically useful because then by measuring AMH circulating in the blood, we could get an assessment of how many follicles were growing. And so that's useful to estimate ovarian age, ovarian function, and ovarian response to stimulation in the context of IVF. So when I joined Pat's lab in 2011, my first task was to make more AMH so that we could study it. And it was very difficult at the time because most of the AMH that we made was inactive. So AMH is a member of the TGF-beta superfamily, and it needs to be cleaved to be activated. So we started tinkering with the protein structure, swapping out leader sequences, optimizing the cleavage site until we found an analog of AMH that we called LRMIS that was optimally cleaved, secreted in high quantities, and then we learned how to purify it and get a high level of purity. So this was one innovation that made some of these experiments possible. The second is we partnered up with Guangping Gao at UMass, who is a whiz at gene therapy, and he helped us produce an AAV construct, AAV9, to now deliver this AMH. And this allowed us to do more longer-term experiments in vivo. And AAVs are very good at infecting both the liver and the muscles, and muscles are very long-lived cells. And so what ends up happening is the muscle is essentially a bioreactor for the production of AMH. So you infect the muscle, and as long as the muscle is there, it's producing the protein, it's going into circulation, and it's affecting the ovary. Now, Pat was working on ovarian cancer, and this was my project. I would collect ascites from patients, try to derive cell lines, try to grow these cells in culture, and xenograft them in mice, and then look at whether they would respond to the gene therapy with AMH or not. And I collected a lot of ascites and tested many different xenografts. And it was very frustrating because sometimes I would get a response and sometimes I wouldn't. And I was really puzzled as to why that was. And it took us 10 years to figure out why. And some of the work from Maëva Chauvin, a postdoc in my lab, showed that actually ovarian cancer cells are actually negative for the AMH receptor. They had no ability to respond to the AMH receptor. Where we were getting our response was actually a contaminating cell line, mesothelial cells. Mesothelial cells share epithelial features. They're very hard to distinguish from other epithelial cells. And it turns out that those were the ones responding to the AMH and in turn affecting the cancer cell growth. And we have many interesting publications that if you're interested in following up on that story, I encourage you to read. It turns out that a cancer-associated mesothelial cell or a progenitor for stroma in the tumor, and they're also antigen presenting and can modulate the immune system and are a terrific target for development of immunotherapies. But that's not really what I'm here to talk about today. I'll go all the way back to September 2013. I've just produced these gene therapy vectors. I'm testing them in the context of ovarian cancer. And there's one particular patient cell line called patient W that's very slow growing. It took 80 days for it to reach the size at which I can treat it with the gene therapy vector. And then it's still very slow growing after the treatment. It takes another 39 days for the controls to reach endpoint. And during that whole time, after treatment with the gene therapy vector, AMH levels are very high in the microgram per ml level. It occurs to me that we had never observed long-term treatment of an animal, of a mouse, with such a high dose of AMH, 39 days at over 1 microgram per ml. And after I'm done measuring all the tumors in the flank, I think, well, maybe I should get a look at those ovaries because as a trained ovarian biologist, I remembered the introduction to my thesis. I had enumerated all the different transgenic mice that had follicular defects. And there's only one paragraph on primordial follicles, and it's the AMH knockout. And the AMH knockout has a little bit excess activation of primordial follicles. And so it depletes its ovarian reserve slightly faster and has a bit of an excess of growing follicles. So essentially saying that AMH is probably a slight break to the system. But this is what I observed. I opened the first mouse and I find that the ovaries are very small. And I've looked at hundreds and hundreds of ovaries by then. And these appear to me the size that you would expect in a newborn mouse. I opened the second mouse. I get the same result. The ovaries are also minuscule. And I instantly realized what had happened. He had underestimated the power of AMH. It wasn't a slight break. In fact, if you increase the level of strong highly enough, you could completely stop follicle development. And this felt like being hit with lightning. I see my life flash before my eyes. I knew exactly what I had to do in terms of the studies that I needed to do. And I basically saw my entire career ahead of me, that this would probably be what I'd be focusing on for the rest of my career. And so that night, I was tossing and turning, thinking about all the experiments that I needed to do. The first experiment that I wanted to do is figure out if and confirm that this wasn't just follicular death, that when I treated with AMH, what had happened is that follicle development was stopped, not that I killed the follicles. And so this I knew how to do very well. I took the ovaries, sectioned them, counted the follicles, and just as I expected, there was a normal number of primordial follicles. They were essentially unaffected. But what decreased are all the growing follicles. There was a massive decrease in primary follicles and essentially a complete absence of all the other growing follicle types, secondary, antral, and even atretic follicles. The second was a little bit harder. Is this reversible? Because for this to be useful clinically, it would be a huge attribute if we could reverse it. But gene therapy is not something that you can stop. Once you've injected it, the muscles keep on making AMH and you can't turn that back off. And so it occurred to me that the only way I could test this was if I would transplant the ovaries. And so I read up, I looked at Stephanie Pangus's protocols, how to transplant an ovary, and I thought, I've got this, I can do this. So I took the ovaries from a mouse that was treated with AMH, and then I put it in a mouse that was treated with a gene therapy for GFP. And we're thinking that now this mouse would be in an environment, this ovary would be in an environment where the AMH would be normal. And then just to control for the surgery, I took ovaries from a mouse treated with AMH and put it in another mouse treated with AMH, presumably to show that those follicles would not get reactivated. And this was easier said than done because it turns out that when the ovary shrinks, so does the bursa. And I needed the bursa to keep the ovary in its place after the transplant. So this is very, very difficult. I had to make a very small incision in the ovary and remove the ovary chunk by chunk and then put the new ovary in, and vice versa, trying to shove a giant ovary in a very, very small bursa. And then I didn't really know how long to wait because, this experiment had never been done. And I thought the longest I can wait is about 2 weeks because I really need to know the answer to this. And I figured by then, probably primary follicles would have popped up. And to my surprise, actually, exactly that happened. Very many follicles started to grow. And in fact, many of them had reached secondary and antral stage by then, which was very surprising how fast that would happen. But essentially proving out the hypothesis that, this stall in follicle development could be reversed. And so equipped with these two findings, then I decided I would share this at lab meeting. And this is actually the conclusion slide from that lab meeting that I showed. I had kept this a secret because I wanted to really prove this to myself and be confident about the results. Because just showing slightly smaller ovaries probably wouldn't have resonated in the same way it did to me. And so this is my conclusion slide. I said, maybe AAV9 LRMIS could be used as an irreversible contraceptive for cats and dogs. Like, keep in mind, this is just 4 months after I had done that experiment. I thought maybe the purified protein could be used as a contraceptive in women. Maybe we could use that treatment to prevent premature ovarian failure or menopause. Maybe we could use it to extend the reproductive window. And maybe we could use it as an oncoprotective agent. And this just shows you how green I was because I didn't even realize that there was a term for this called oncofertility. Teresa Woodruff had already coined this term. But essentially the idea that could we preserve fertility when we treat with chemotherapy, like cyclophosphamide or cisplatin. And so essentially, 4 months into this work, I basically mapped out my entire career. These are the exact same things I've been working on for the last 10 years. The other reason why this was memorable is I also announced to the rest of my lab that I was going to be a father, and this is Isaac. So then when I was promoted to assistant professor in 2016, I started working on this in earnest. My lab was going to work on the role of AMH in the ovary. And so the first question I wanted to look at is, I knew this was reversible, but the results were kind of surprising to me, just how fast those follicles were returning. So I thought I would do this in a much smarter way than, doing this harebrained ovarian transposition. I would treat with the recombinant protein. By now, I knew how to make a lot of it, and you need to inject it every 12 hours. So this is a long experiment. I treated for 40 days because that's how long it took for in that initial experiment with patient W. And then I stopped treatment and I asked the question, how long does it take for this follicles to return? And to my surprise, it only took 5 days for primary follicles to return to normal. It only took 10 to 15 days for secondary follicles. It took 30 days for antral follicles. And just as secondary and antral follicles came back, you can see AMH came back because those are the ones producing the AMH. So this was much faster than I expected, because what I would have expected from the literature was that it would take 2 to 3 months, anywhere up to 6 months. These are the estimates that I found at the time. So actually, follicle development can be quite rapid. The second question was, well, can these follicles ovulate? Are they competent? And we had noticed that actually when we stopped treatment, this wave of growing follicles was actually a little bit larger than normal. We had actually an excess of antral follicles by day 30. And so, we could test two things at once. Maybe we can test whether these follicles can ovulate, but also could we get a greater yield of superovulation? This is a common technique that's used for assisted reproduction. And so to test this, we stimulated with gonadotropins at day 15 post-release and at day 30 post-release. And just as you'd expect, at day 15, you don't have antral follicles, you don't get a response, you don't collect any eggs. And then you stimulate at day 30, and then all of a sudden we got 3 times as many eggs, which is even greater than what we were expecting, because we had maybe a 60 or 70% increase in the number of antral follicles. So not only did we have more antral follicles, but actually they were hyper responsive to the gonadotropins. And this is the technology now that's been licensed and is in clinical development by Granata. Next, we wanted to know how this works. And then I had heard rumors of this great technology that was being developed at HMS, where they would use microfluidics to package cells and droplets with barcodes, and it sounded very interesting. And I had some of my friends telling me how this would revolutionize biology, and I thought this may be the right tool that we need. And so we did an experiment. We learned how to disaggregate ovaries. We tried many different reproductive states. So we took ovaries from mice at different stages of the estrous cycle: proestrus, estrus, metastrus, diestrus. We took postpartum mice, lactating, non-lactating, and then just the non-monitored mice to see if we could match them to the right cycle stage. And then, sure enough, when we analyzed this data, we found that there was actually massive changes in transcription and cell states across the cycle. And we even found many, many different secreted factors that we could detect in the blood that would change in a cyclic fashion. Then we proceeded to sit on this data for 5 years until the right person came in to finish this work, and that's Marie Meinson. You'll see this is a kind of a recurring theme in a lot of those studies. And so we had the right tool. Now we wanted to apply it to the ovary and to AMH treatment. The problem was that in these adult ovaries, we basically didn't recover any primordial follicles, because although they're quite numerous, they're very small. So on the percent cell basis, they're actually very rare in the ovary. So we thought the best way to do this then would be to use a fetal ovary. And so we know AMHR2 is expressed very early on, even before follicles were formed, in the cells that will become pregranulosal cells. And then in mice, follicle development actually occurs postnatally. So between postnatal day 1 and 3, you get cis breakdown, follicles are individualized, and you get your first wave of follicle growth. And then by postnatal day 6, you start getting secondary follicles. So we thought if we inject them right when they're born, it takes a couple of days for the gene therapy to work, for AMH levels to rise. We'll look at them at postnatal day 6. By that time, they've grown, and we'll be able to both see the effect on primordial follicles and the effect on these early growing follicles. Perfect plan. Then I recruited the right postdoc to do it, Duygu Satsioglu, and she had been studying actually the follicle awakening on the oocyte side. So she was an oocyte expert, and I thought with my expertise in granulosa cells, together we'll figure this out. And so we had the protocol ready. We were ready to do a single cell RNA-seq. And then she comes to me and says that something is weird. The uterus don't look normal. I'm like, what do you mean? We've been looking at hundreds of mice that we treated with gene therapy. The uterus always looks fine. Whenever we treat an adult mouse, we've looked very comprehensively. AMH does nothing to the uterus. But I looked at those tissues, and sure enough, she was right. The uterus was really, really thin and small. I thought, and I said, Duygu, I'm sorry, but now we're studying the uterus. This is what we're going to be working on. Now we're a uterine lab. And so not only did we do the ovary in that experiment, we decided to do the uterus at the same time. And this is actually one of my favorite stories because it brings back such a cool aspect of developmental biology. So I'll bring you back to what I explained at the beginning with Jost, in that AMH is produced by the fetal testis, and it causes regression of the Mullerian ducts at the time at which the urogenital ridge is bipotential, meaning it has both male and female ductal structures. And all it's waiting is for the signal from the gonad, so the AMH from the testis, to activate these cells that you see here around the Mullerian duct to cause regression. And then it was always thought that this was a very short window of development where you need to receive the signal and you regress. And if you don't receive the signal, then the Mullerian ducts develop and you do your female sexual development. But nobody had ever asked the question, what happens to these cells in the female when they don't receive the signal? And in fact, we see these cells, they perdure. We find them at E18.5. We find them at E19.5. We find them at birth. We even find them at postnatal day 2. And it's only at postnatal day 6 do they disappear. And that's precisely at the time at which follicles reach secondary stage, at which they start making AMH. So never the two shall meet. This is such a beautiful example of developmental biology. The uterus is sensitive to AMH right until the day at which the ovary starts making AMH, because if it remains sensitive, then it would regress, and then you wouldn't get urinary development. And the single cell data was as clear as a crystal. We could see the progenitors being inhibited by the AMH, and what it did is it prevented those cells from becoming inner and outer stroma. It turns out that the very cell that in the male causes regression of the Mullerian duct is the one that in the female builds the uterus, builds the endometrial stroma. And we, by chance, had captured a very precise developmental window in which these cells are specified. And so this is what happens here. So initially, the treated uterus looks the same between control and AMH. But in the control, the uterus develops, you form those different layers, the endometrial stroma develops and grows. But in AMH treated, this progenitor is inhibited. It's prevented from proliferating, it's prevented from differentiating. And the effect is so profound that actually, if you just treat with the recombinant protein for a couple of days between birth and postnatal day 6, the uterus never recovers. You can look at the uterus a year later, it's still basically an empty shell of myometrium. And so we had identified a very precise developmental window in which the endometrial stroma is specified. Then we had to go back to that ovarian experiment. And once again, Marie Meinson had to finish a study. And what we want to know is exactly how does AMH suppress follicle activation and development. And once again, single-cell RNA-seq proved to be key in understanding this picture. So just to orient you, in control ovaries, and this is granulosa cells from Parmagel follicles are quiescent. And then they get activated, they start differentiating in granulosa cells, from progranulosa to granulosa, and then they enter mitosis. So the follicle starts to grow. But when we treat with AMH, we get a completely distinct transcriptional state where you find our two bottlenecks. So the quiescent cells are hyperquiescent. The signature of quiescence is really elevated. So they're having a hard time activating. And those that are activated are not differentiating. They're stuck, they're paused, and they're never entering mitosis. And so so they're blocked from further development. And this is visualized here. If you look at Ki67, the marker of proliferation and control, you find these follicles that are highly positive because follicles grow very rapidly. And the AMH treated, the follicles are negative. And then likewise, NR5A2 is a great marker of an activated follicle. And as soon as the follicle activates and starts to grow, it starts expressing NR5A2. And the follicles, even the ones that appear to be activated, are failing to turn on the proper transcriptional machinery. And we've continued to work on this and trying to understand what does it mean exactly to be quiescent. And this is actually a question that we often ask ourselves in the context of cancer, because this is a way of cancer cells to evade, for example, chemotherapy. It's basically you exit the cell cycle, you enter G0 or G0. And this cell cycle exit is regulated by the CYP-KIP family of CDK inhibitors. And just like in cancer, this is exactly what we observe in the follicles. When we treat with AMH in the granulosa cells, we see massive upregulation of all three CYP CDK inhibitors, P21, P27, P57, and their downstream targets are dysregulated RB1 and E2F1, basically signaling cell cycle exit. What's surprising is that we actually observe the exact same signature in oocytes. So that's surprising for two reasons. One is that the oocyte actually doesn't have a receptor for AMH. That means that this signaling must be communicated between the granulocyte cells and the oocyte, so that both essentially induce quiescence. The second is that the oocyte is not a mitotic cell. The oocyte is arrested. And so why does it need to induce CDK inhibitors? That is still a mystery. And so again, we tested for reversibility of this effect. We did this experiment in neonatal mice. And if we treat for 10 days and we release, we could see that the CDK inhibitors basically come back down to normal within 5 days. And that's coincides with reentry in the cell cycle. So we see upregulation of TOP2A by single cell RNA-seq, and by immunohistochemistry, we see Ki67 coming back up. And the cell cycle exit is one aspect of quiescence, but there's actually quite a few other ones. One, as I've already shown, is inhibition of differentiation, and we do see that. But one is increased DNA damage repair. And this is something that intuitively makes sense. If you're going to have a follicle arrested, maybe you have 50 plus years, and if this follicle is very, very precious because you only have a limited number of them and you need them for reproduction, then you better make sure that you maintain genomic integrity. The second thing is we've noticed that this quiescent phenotype is associated with a lot of secreted factors, and we call this a quiescent-associated secretory phenotype. Many of these factors are actually reminiscent of senescence. The difference here being that this is a reversible state. But there are examples of reversible senescence that occur in development, and even in tissue repair, like liver fibrosis. And so the question is, what are these secreted factors doing? Are they communicating a message to immunocytes? So many of these are chemoattractants. Are they communicating to other follicles? Are they impacting the stroma? And we have a really mixed bag of signals here. Some of them are chemoattractants, others are immunotolerant messages like TIM3 and CXCL14. But I'll come back to the DNA damage in a second. So next, I'm going to talk about oncofertility. So the concept here is quite simple. Chemotherapy is very damaging to dividing cells. Follicles are actively growing structures. They're highly mitotic, so they're very susceptible to chemotherapy. And then these growing follicles are actually also the source of AMH, and AMH acts in a negative feedback to the activation and early growth of follicles. So you lose follicles both ways. You lose follicles because of toxicity from the chemotherapy, and you lose follicles by excess activation. And the idea initially there was quite simple: is can we pause follicle growth, make the follicles less susceptible to chemotherapy, and can we enhance the feedback and make sure we don't get an excess activation of parenchymal follicles? And we did this experiment very early on. We used osmotic pumps implanted in the abdomen of mice that were eluting AMH concurrently with 3 different chemotherapies: carboplatin, doxorubicin, and cyclophosphamide. In all 3 examples, we showed that we could maintain the ovarian reserve. We had a significant preservation of parenchymal follicles with doxorubicin, carboplatin, and cyclophosphamide. What struck us is that this was particularly effective in the context of doxorubicin. So when we wanted to look at mechanism, we thought, well, let's focus in on doxorubicin. This is where we're getting our best productive effect. And this is the work done by Fong and Ilana. And again, use our favorite tool, single-cell RNA-seq. We treat the mice with doxorubicin concurrently with AMH. Take the ovaries 4 hours after the second insult with doxorubicin and look at what is the effect of doxorubicin on all the different cell types of the ovary, and what's the effect of the AMH rescue. And unsurprisingly, we found a very pronounced P53 response. Of course, you generate DNA damage, you get a P53 response. But somewhat expectedly, we find AMH attenuates this P53 response. So why P53 response? Of course, when a cell senses DNA damage, the first thing it has to do is cause cell cycle arrest, because the last thing you want to do when you have DNA damage is replicate, right? You want to give some time to the cell to repair its DNA. And so P53 will induce both cell cycle arrest and the DNA repair pathways. And if it can't repair the DNA, then it'll undergo apoptosis. And there's a number of checkpoints and feedback mechanisms to ensure that this is well regulated. And when we looked at evidence for this, in the ovary, we found basically evidence for both protection by quiescence, so we're preventing proliferation, but also it seemed that we could find evidence for enhanced DNA repair, which was really quite unexpected, not exactly what we had expected, because AMH is a hormone. So what does that have to do with DNA repair? And this is very brilliant work by Eunmi Chang, and she noticed that actually, if you looked at early follicles, basically primordial follicles and primary follicles, which are non-mitotic, this is where we actually observed the best protective effect. So this couldn't be because we had prevented proliferation. This had to be another mechanism. And so we quantify DNA damage by basically looking at gamma H2AX, which is a phospholipid alternative histone. And we find that gamma H2AX is greatly elevated with doxorubicin, and it's reduced with AMH. And I thought, how is this possible? How is a hormone, enhancing homologous recombination repair? Because that's what doxorubicin is inducing, is double-stranded breaks. And Eunmi came back with two papers, and she said, actually, one of the prime targets of AMH that we use routinely in the lab, actually to measure response to AMH, is induction of ID3. ID3 is a DNA binding factor that plays a role in transcription, but also it turns out plays a role in the loading of the DNA repair machinery, and particularly the homologous recombination repair machinery, and such as MDC1. And so what we thought was happening is maybe AMH was inducing all these DNA repair genes, and they were facilitating the resolution of double-stranded DNA breaks. And so we tested this hypothesis using siRNA against ID3. And just as you would expect, if you give doxorubicin, so this is now primary cells in culture. If you give doxorubicin and you look in the nucleus, you find all these foci of double-stranded DNA breaks. And if you give AMH, these foci get resolved and you find fewer H2AX foci. And instead, you find this speckled pattern of ID3. And if you downregulate ID3 by siRNA, and so you've got AMH rescues and reduces the number of H2AX foci. If you downregulate ID3 by siRNA, you lose this effect. So meaning that the repair mechanism was at least in part due to upregulation of ID3. This is all nice and fine. We've got a mechanism, but is it biologically relevant? And so we tested this. We give this same treatment in mice, in a cohort of mice, give an osmotic pump with AMH, treat with 2 rounds of doxorubicin, and then we put them in males and we followed them for 6 months looking at their reproductive output. And sure enough, basically their fertility was doubled. And so not only were we preserving follicles, but those follicles were competent and they could give rise to litters. Next, I want to talk about another application, which is contraception. And this one is quite obvious, right? This was the first thing, of course, that popped in my mind. If you don't have any follicles growing, of course you can't ovulate. Of course you're contracepted. What's interesting about this is that when you treat a mouse with AMH, with gene therapy with AMH, and you immediately put it with a male, it can actually still produce litters. It can still reproduce for about a month. And it shows you that, in fact, AMH doesn't prevent ovulation, and it doesn't prevent fertilization. It doesn't prevent embryo development or implantation or fetal development or parturition or any process in reproduction. What it prevents is the replenishment of growing follicles. And it takes about 4 weeks to basically void the ovaries of growing follicles. And then they're not replenished by new growing follicles. And the animal will then be contracepted for the rest of its life. And this we thought would be great to use as an alternative to surgical sterilization. And this is work done by Philippe Godin, who incidentally is starting his own lab in a couple of months and will continue to work on this. And the idea was we would take this transgene, target it towards the cat, and then use gene therapy then in cats and test whether we could prevent reproduction in cats, in female cats. So why do this in cats? So it turns out there's a great need for alternatives to surgical sterilization. There's over 480 million unowned domestic cats worldwide, and the way we handle this overpopulation is through TNR, trap, neuter, and return. You have to capture the cats, neuter them, and then usually notch the ear, return it back. And this is very resource intensive because you need then a surgeon and post-operative care and a lot of technical skill to be able to achieve this. And this is basically fighting a losing battle because cats are very, very prolific breeders. They'll have multiple litters annually, and they reach sexual maturity very early on, within a year of age. And not only is this a humane concern for cats, but it also is an environmental concern as well. Just in the United States alone, free-roaming cats cause the death of over 2 billion birds a year, and they've contributed to the extinction of over 33 species. So all this to say that there's a great need for surgical alternatives to sterilant. And so we've done many different studies, and I'll just show you one example here. This was done in adults. And we published this in 2023, Nature Communication, where we treated mice with a control empty vector, a low dose and a high dose of AAV9 EMH. And then we monitored these cats, we monitored their hormones, and then we did 2 fertility trials, one at the 1-year mark and one at the 2-year mark. And you can see we continued to monitor these cats for many, many years, and they've maintained very high levels of AMH, 100 to 1,000-fold higher than you'd find endogenously, and presumably equating with lifetime contraception. And so we've looked at the safety of this approach. The injection sites were monitored, everything looked good, physical exams. We even treated kittens and showed that it doesn't disrupt their development, and they can still go through puberty. And we've done multiple studies, and so far we've tested this in 19 cats. And I'm happy to say that none of them have ever gotten pregnant, and all 8 controls that we treated with the control vector today have gotten pregnant. So far we have a 100% success rate. And we've learned a lot about the mechanism of action and cat reproduction. So cats are a little bit different than many other mammals. They're induced ovulators, meaning that they actually have to copulate to induce the ovulation. That's what triggers the LH surge and the ovulation. And you can follow this through progesterone secretions. And then we know for a fact that our cats can't ovulate because we follow them as they would mate and then look at their progesterone in their blood over time. And normally what you would find in a control is as they mate, it would trigger ovulation, progesterone would rise and stay up throughout the luteal phase. And then in our treated cats, those that mated, would continuously mate, but never trigger an ovulation. So the progesterone would never rise. So we know our cats did not ovulate. Last, I want to talk a little bit about premature ovarian failure and mostly menopause. And I want to show some preliminary data that I find extremely exciting. But you have to take it with a grain of salt because it's very preliminary. And I feel as though we've been struck with lightning again, because this basically made me reconsider everything I thought I knew about ovarian biology, about ovulation and follicle development, about hormones, and what is the fundamental nature of menopause. So recently we started getting ovaries back from those cats, and they look exactly like you would expect. In fact, they look exactly like the mice that we've been treating, in that they basically had no growing follicles. They still had primordial follicles, but if anything, they were even more suppressed than what we had found in mice so far. We basically, in many cats, could not find any follicle beyond the primary stage. And so, of course, these cats never got pregnant. But here's the problem. We had also been monitoring the hormones in these cats. And when we looked at pre-treatment and post-treatment, the hormones were all normal. Testosterone was unchanged, estradiol was unchanged, and inhibin B was unchanged. And we all know from textbooks that you need follicles to make hormones. So one of these two things had to be wrong. They couldn't both be true. And not only were the hormones levels maintained, but so was the estrus cycle. These cats were undergoing estrus. They were mating with males, and they wouldn't do that if there was no estrogen. To tell you the truth, this had been a thorn at my side for over a decade because, basically I'd observed more or less the same thing in mice. we know these mice can be contracepted basically for life. We know they essentially have no growing follicles. There's a few secondary or antral follicle here and there. But yet, when we look at the estradiol levels are maintained. And I always try to explain that away. Well, maybe this is the ELISAs for estradiol are notoriously unreliable in mice. So maybe this is just phantom data. And then but what about all those mice where we don't observe any growing follicles, but I can still detect estrus when I look at vaginal swabs where there's signs of estrus. So how can that be if they don't have estrogen? Right, and so if this is to be true, then hormones have to be coming from somewhere else other than follicles. And this is such a fundamental concept of reproductive biology, is the 2-cell 2-gonadotropin hypothesis. Right, so we all know this from textbooks. Theca cells have CYP17A1, they make testosterone, and a granulosa cell in the follicle has CYP19A1. Aromatase, and it transforms testosterone into estradiol, and this is how you make estrogen. This is what the ovaries looked like in those cats. They had no growing follicles, yet they had CYP17A1 and CYP19A1 diffuse stain all throughout the ovary. So this is kind of a smoking gun. We're looking for where the hormones come from, and we find the right cells right there in front of our nose in the ovary. It just didn't look like anything unless you stain for it. And so there's a Theca-like cell that produces testosterone, presumably, and a granulosa-like cell that can aromatize it and produce estrogen, presumably. And these two cells are distinct, and we know because we can do a dual stain at the same time, and we find this mosaic pattern. They're always next to each other, which you need to do the steroid conversion, just like you have in a follicle, except there's no follicles. They're all throughout the ovary in this way. And it's not just steroids. Actually, these same cells that are CYP19A1 positive are inhibin B positive, so they can make other protein hormones. They can basically recapitulate the follicle without being a follicle. They're also in part MHR2 positive. So the next question was, well, maybe this is just a weird thing that cats do. Maybe cats are unique and they can make hormone without having follicles. But actually, we know mice are more or less age like humans. We sometimes dismiss this, but mice can live 2 years. And if you look at mice at 12 months of age, 1 year of age, they're essentially becoming infertile. They have almost no pharyngeal follicles. They have very, very few growing follicles. They're essentially a perimenopause model. And if you age these mice further and you reach 18 months of age, they're completely devoid of follicles. And so we thought, well, this would be a great model in which we could test our hypothesis of whether we can induce follicular mimicry in the stroma of ovaries. And we did exactly that. We aged mice to 12 months. We treated them with the gene therapy. And, again, take that with a grain of salt because we have only a few number of mice. We observed the exact same pattern. There was diffuse staining in the stroma of both CYP19A1 cells and CYP17A1 cells, suggesting that we were able to recapitulate this phenotype of stromal hormone production in mice. So I was talking to a colleague at Oxford and describing, our phenotype to her, Cecilia Lindgren. And I was telling her, our cats basically have the opposite of menopause. And without missing a beat, she tells me, you mean menago? And what I'd like to propose here is that, for even in our own case, since the beginning, what we've been trying to do is, try to preserve the oocyte, try to combat DNA damage and oxidative stress, and try to prevent follicles from activating, try to protect the genomic integrity of granulosa cells, try to slow down follicular growth. But actually, maybe this is a red herring. Maybe follicles don't matter as much as we think if we want to, induce menago. So what I would suggest is that if we can understand what we've just observed in mice and in cats, if we can understand how the stroma is able to compensate for the lack of follicles and produce hormones, then maybe we would have a way to cause menago in humans and maybe avert all the side effects of menopause and prolong a healthy health span. And so now I'm suggesting that for the next decade, this is probably what I'll be working on. So with that, I'd like to thank all the people in the lab that have contributed to this. I have a terrific team. And there's many alumni and essentially everybody in the lab is working on some aspect of this, whether they know it or not, because this is so fundamental to ovarian biology. And I have many collaborators. I've shown pictures of them previously at CRU and Thompson Lab, at UMass, Landa Lab, and many, many others that, I can't cite all of them. And I particularly also want to thank the funding that I've received, and especially those two at the top, the DoD Ovarian Cancer Academy and the Michelson Found Animal Foundation, particularly because they believed in me very early on when I was setting up my lab and invested both in the ovarian cancer projects and then the reproductive projects around AMH when I was really struggling to convince anybody that AMH was important in the ovary and that I thought had, really great potential clinical applications. And of course, we've received many other sources of funding since, including from the NIH, Bill and Melinda Gates Foundation, and many other founding bodies. So thank you.
We have one question in the chat coming from Rajiv Chandra. David, where do you think the mimicry is originating from?
So of course that's the $1 million question. Where are these cells coming from? Are they adopting this new state? Obviously, this is something we're working on. I don't have any proof of any particular lineage so far, but we're actively working on this. We have some ideas, but it would be pure speculation.
Next question. We know that AMH receptor 2 is not expressed in primary follicles. What is the path explaining how AMH maintained the early stage?
So that actually AMH2 is expressed in primary follicles and primary follicles and all follicle types. And so I showed that earlier in the talk. So the pathway is basically AMH is binding to AMH receptor on granular cells of primary follicles and presumably inducing this quiescent state.
Next question is about Menogo. For the Menogo work, were you able to rejuvenate the vagina and see the production of cornified cells if you do vaginal swab? Exciting work.
So actually, we've always, even since our 2017 paper reported this, that we can find cornified epithelium in mice that we've treated with AAV9 AMH. This is persistent. They have estrus-like vaginal epithelium throughout their life.
Question in the audience.
Hi, David. I've known you a while, and I will hope to see you at Indo, but I looked at their presentations, and obviously I think SSR has these too, but there's obviously a big focus on PCOS and a lot of ways to treat, target, sort of understand the etiology of PCOS, which one of the markers now, or one of the ways you can diagnose it, is elevated AMH levels. But it seems like your contracepted mice and cats don't really have that same sort of pathophysiology. So if you could just talk about, what do you think the intersection is between these two mechanisms, how they might be different, how they might be related?
Yeah, so this is a fascinating conundrum, right? Because every time I present this data in a reproductive conference, everybody's like, well, are you inducing PCOS? In fact, I'm inducing the exact opposite of PCOS because there are no cysts and there's no elevated androgens. And so I think it's telling us something fundamental about PCOS, that presumably that phenotype of PCOS is probably not, those particular phenotypes are not probably driven by excess AMH. If anything, maybe it's the opposite. Maybe it's resistance to the AMH signal, right? And we do know that at least in some occasion, there's been reports of AMH mutations in AMH or AMH receptor in patients that have PCOS. And perhaps some of the PCOS phenotypes, such as the annual ovulation, maybe that's related to AMH, but certainly we've never seen this in any of our models. And the ovaries from our cats are pristine; they're very small, and they don't have cysts.
So I just want to say congratulations again to Dr. Pepin for his very inspirational and insightful work and being dedicated to your ideas, and from a very early age following those really scintillating findings to produce a whole body of work. So congratulations again. And I just want to close by saying that on March 13th, we'll be opening the fourth opportunity to be a new investigator with the Roger Short Medal and Lecture. And we would encourage all young investigators to look into that to see if they also want to consider being nominated for this very prestigious award. So thank you again on behalf of the three societies, and congratulations.
Thank you. Oh yeah, so there's a reception afterwards in the corridor. So I'm hoping all of you can come.
Congratulations.
Thank you. Thank you so much.